So let's get into the steps. So we kind of explained how, the action potential came running down the T tubules deep throughout the muscle. And then we kind of jumped to calcium being released from the sarcoplasmic reticulum. So let's kind of fill in the gap here of how that action potential running along the T tubule results in the rushing of calcium out of the sarcoplasmic reticulum and into the sarcoplasm. So the SR is gated by these channels, right? They are voltage gated channels. And then the channels are also coupled to ryanodine and a DHP receptor, okay? And so if we look closely here, we can see the T tubule. This is the wall of the T tubule here in green. We've got our two receptors here. The receptor in purple is the DHP receptor. The coupled receptor in green is a ryanodine receptor, right? These are voltage-gated receptors. And so when the action potential gets down into this invagination, it's going to change the voltage of the membrane. That's going to trigger the DHP receptor to then pass the signal along to the ryanodine receptor. And the ryanodine receptor is also a channel, right? Similar to our ionotropic receptors, the ryanodine receptor is going to open up the calcium gates that allow calcium to move from the SR. So this region over here is representing the SR. It should be shown here in blue, the way it is here. And then it goes from the SR, as we can see in this state, into the white region here, which is really the cytoplasm of the muscle cell, okay? So this is how we get that calcium from its storage area out, flooding the cytoplasm so that it can be in contact with the troponin complex, and it can initiate the exposing of those sites and start that cross-bridge cycling. And then we said that in order for this process to start, the action potential induces those channels, but then these channels are also closed by the excess amount of calcium. So calcium is going to induce the opening of these channels. It's also going to induce the closing of these channels. So when there's too much calcium flooding the membrane for too long, that also triggers up the closing of these channels so that we can kind of, in a negative feedback type of way, close the channels and prevent more calcium from being dumped into the sarcoplasm. All right, and just to reiterate this idea here that in order to terminate or to cease muscle contraction, calcium must leave troponin. In other words, it must leave the cytoplasm or the cytosol. And the way that it does that is it's pumped back into the SR. So we've got calcium ATPase pumps to actively pump calcium back into the sarcoplasmic reticulum in order to bring about the relaxation of muscle. We also have so we got the pumping of calcium, and it's also important that the rate of the pumping of calcium into the sarcoplasmic reticulum also translates to the rate at which we can generate another muscle contraction. And we'll talk about that later on, but if we can get those calcium pumps to clear the cytoplasm really quickly, then that will actually increase how fast we can generate another series or another sequence of muscle contraction. And we'll talk about how the rate of the calcium ATPase pumps plays a role in the rate of muscle relaxation and muscle contraction. Okay. What questions do we have? So let's kind of pause here. That's objective 2, which is quite a lot. So all of cross-bridge cycling, all of excitation contraction coupling, and then how calcium plays a role in regulating this process, turning it on and turning it off. Okay. So I don't think we'll go any further here. On Monday, we'll probably start off with the twitch. And we won't get very far into the twitch. We'll probably just complete just the twitch and the 2 types of twitches, which is just a couple of slides. So probably about the first 20 to 30 minutes or so of class. And then we'll reserve the rest of that class period for questions you may have and for any additional review. Okay, so someone's asking to go over where that calcium comes from. Okay, so I'll go back here Oops. Okay. So here we can see that the calcium is stored in the sarcoplasmic reticulum. So that's the organelle in the cell that's storing calcium. And I just want to point out why it looks the way it does. So we talked about the fact that muscles are pretty much packed end to end with these myofibrils inside the cells. And so this schematic is really not doing justice. It should be showing the muscle fiber with those myofibrils kind of jammed in there. And the sarcoplasmic reticulum is really wrapped around the periphery of the cell, and it's right underneath the sarcolemma. So let's actually go back, way back to one of the earlier slides here, where it kind of showed that a little bit better here. There we go. This one. So we can see that the sarcoplasmic reticulum is really kind of wrapped right underneath the sarcolemma, wrapped around the periphery of the muscle. And we've got all these myofibers kind of just jam-packed in the muscle fiber. And this is where we want that the calcium to end up in order to make interactions with the proteins on actin, right? So we've got to get the calcium that's being stored here out into the cytoplasm, the sarcoplasm, so that it can interact with the actin that's on these myofibrils. So just kind of bear that relationship in mind. So then we've got the binding of that calcium. Here we go. So then we've got the movement of that calcium rather from the sarcoplasmic reticulum out into the sarcoplasm in order to start this whole process. And so we can pick up from the events of the action potential getting down into the T-tubule. Let's go to this image, because I think this really helps to clarify how we get from an action potential through the T-tubule to then the release of calcium from this structure. So because the T-tubule is so closely connected with the lateral sacs, remember this is a triad happening here. This is our T-tubule in the center, and the lateral sacs or the terminal cisternae on either side. And this is why they have to be in such close proximity. They're actually being connected via these two receptors, the DHP receptor and the ryanodine receptor, in order to couple this process together. So when the action potential excites the membrane of the T-tubule, that has a voltage change on the DHP receptor. The DHP receptor is pressed up against the ryanodine receptor. So it's going to kind of send that signal to the ryanodine receptor. And so that's going to trigger the opening up of that receptor, which is also a channel. And so calcium can move from its storage area in the SR out into the sarcoplasm where it needs to be in order to interact with those regulatory proteins, specifically troponin, to expose those binding sites and initiate that process. Okay, hope I kind of cleared that up. So it's really important to kind of visualize how all this is taking place, the importance of the triad, the importance of having those storage areas for calcium so close in proximity to the T-tubule to spread that electrical energy to then result in the release of calcium to start this process.